E.coli Identification Project Using AI Assistance by Jailyn Marquez and Anthony Benvenutto. Introduction. The identification of unknown bacteria is essential in microbiology for disease diagnostic, environmental monitoring, and understanding biodiversity correctly. Identifying bacteria helps scientists understand how they cause disease, respond to antibiotics, and function within their ecosystem. In this experiment, various biochemicals and staining techniques were used to identify an unknown bacteria specimen labeled as unknown number 9 and number 21. These methods include gram staining, selective and different media, and metabolic test. The goal of this project was to isolate and identify the unknown organism using systematic laboratory methods and compare the results to known bacteria characteristics. Abstract. In this experiment, we tested an unknown bacteria culture that was identified by gram staining and biochemical testing. The organism appeared as gram negative and formed smooth off-white colonies on nutrient agar. A series of biochemical tests, including lactose, sucrose, and EMB agar growth test, was performed to determine metabolic characters. The sample test did positive for lactose and sucrose. It showed a metallic green shear on EMB agar, further proving it is E. coli. Based on these results, unknown was identified as E. coli. The result was supported by its consistent biochemical profile and its characteristics growth patterns on selective and different media. Methodology. The unknown culture was first streaked into agar plates using a streak method to obtain isolated colonies. A gram stain was performed to determine cell morphology and gram reaction. Based on the gram negative rod morphologies, selective and different tests were chosen. Results from each test were recorded and compared to reference identification tables. Final identification was made by matching the biochemical profile to known results for E. coli. This is the process that we did to determine if we had E. coli or not. So we had a paper on to help us know what test to do next. So step 1 is where our unknown was. So number 9 and number 21 was in these mini testing tube. And then number 2 and 3 are the gram stains. As you can see, that are like a little off-white, pinkish and everything. So then we did sucrose and lactose test. As number 4 is, as you can see, the color is yellow, which means it was a positive. And then in number 5, that is what E. coli looks like under the microscope. Results. The unknown bacteria were identified as gram-negative rods by gram staining. Colonies appeared smooth, circular, and cream to off-white on EMB agar. Growth production, a metallic green shear, indicated vigorous lactose fragmentation. The organism tested positive for iodine and clacid and negative for crazy. The results matched the biochemical characteristics of E. coli. Additionally, the bacteria showed rapid growth in nutrients broth, suggesting high metabolic activity. Microscope examination revealed rods with uniform sizes and shapes consistent with gram-negative bacteria. Further testing confirmed the bacteria's ability to fragment glucose and prussic acid. Conclusion. Through careful observation of colony morphology, gram staining, and a series of biochemical tests, the unknown bacteria samples designed as unknown number 9 and number 21 were successfully identified as E. coli. The presence of positive lactose fragmentation and iodine test results, along with the characteristics metallic sheen on EMB agar, provided crucial evidence confirming this identification. This experiments demonstrated how different media paired with biochemical arrays can work in contract to achieve precision bacteria identification. Employing multiple tests provided a robust foundation for accuracy and highlighted the importance of the systematic logical approach in microbiology investigation. Further research and acknowledgement. Further research in our own E. coli isolates could indicate testing for antibiotic resistance using molecular identification methods of studying growth under different environment conditions. I would also like to acknowledge my instructor for their guidance throughout the project during the laboratory work. Thank you.